This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1081, recorded on January 19, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. It stopped snowing, but it was snowing 20 minutes ago. The weather is atypical for winter in the Northeast because we're not used to snow anymore. I remember a year where we had over 17 snowstorms in a winter, and then we went 677 days or something like that without any snow whatsoever. So if you're doubting that climate change is here, just go back and check the record.
Dix, how many years ago was that?
Maybe 15 years ago.
And did you walk to school in the snow uphill 30 miles and all that shit?
Both ways.
Both ways.
In your father's pajamas.
That's right. That's right.
Minus 2 degrees. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 47 Fahrenheit and sunny. Tinkerbell's around here somewhere. Oh,
yeah, Tinkerbell's out here. Yeah, there it
down
is.
below
there
my shoulder.
Is.
Yeah, she'll show up. Okay, so it has been cold here. We've had several consecutive nights of sub-freezing weather, cold for Austin. Okay, so Jack Frost has come through and wiped out all the foliage in the garden, which I was fully expecting. It'll be time for me to get out and harvest that stuff and ready the place for spring and new mulch and the rest of it. So that's all fine with me.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone. It is 28 Fahrenheit here, and it is still snowing lightly, and I think it's going to be picking back up.
And from Montreal, Canada, Angela Mingarelli.
Hello, great to be here. So the fact that Rich is even talking about spring, it seems so far away because the snow just started here. We are at minus 15 C, but as a true Montrealer, we always speak with the wind chill. So it's minus 20 with the wind chill, which is minus 4 F. So the fact that Rich says that it's kind of cold at 7 degrees C, I care to disagree. I wish it was 7 degrees, Rich. It's probably 7 degrees in my kitchen right now.
Yeah, I can do a lot of things, but I can't help you with that. You have to talk to McFadden. This is all his fault.
Angela, did you grow up in Toronto, I think? Is that right?
Ottawa, the capital.
Ottawa.
Yeah. Mm-hmm.
So,
also very cold.
Because I just got the boot. You said a boot.
No, I didn't.
Yes, you did.
No, I didn't. What? I say about, not a boot. I don't say
No,
a
you
boot.
just said a boot a few minutes ago. It's on tape. You can hear it. But that's why I'm asking you, because I never heard you say a boot before.
What?
And is that something you would develop in Ottawa? That's what I wanted to ask.
I mean, people think the stereotype is that you say it all across Canada. But
okay, even Western Canada.
Yeah, we're not really Western Canada. Western Canada is like
you're definitely
BC.
Not
We're like central, east. Yeah, I guess.
Yeah, but in Western, do they also have those accents as well in BC and all that?
No.
No, probably not as much. But I mean, in the prairies, I've heard people in the prairies, they have like a very specific
Anyway, you
accent.
Play back that first minute or so. You'll hear you say about, and that's
I'll have to fact check.
I've never heard you do that before. I wondered if Montreal is getting to you,
Maybe it's the French.
Maybe it's the French.
I wanted to ask all of you, all four of you, are you excited to do TWiV every week, or is it a chore?
It's the highlight of my week.
Yeah. No, I'm
I
excited.
look forward to it. I look forward to it. I really do.
I do too. It's not a chore at all, and once we start, it's great.
I sit down and read the papers, and I think to myself, if it weren't for TWiV, I wouldn't be doing this.
No, of course not.
All true.
The papers are like the first one today. The snippet is bizarre. Okay,
Yeah,
it's really great. And
Yeah,
that's
so
right.
that's good. And I think I probably said this before. When I retired, what I was afraid of missing more than anything else, or, whatever, okay, was being able to hang out with my buddies and talk about stuff. And here I
Yeah.
am.
Here we
This
are.
is good. I look forward to being in the same virtual room with you guys.
Here.
All right, good. I'm glad to hear it. I just wondered.
Hey, if I didn't
no
want to do
one's-
it, I wouldn't.
Yeah,
I was going to
understand.
say, yeah, no
retired.
one's forcing us to be here. We could just not
No.
come, Vincent.
I asked you if you wanted to do it, and you could say, but no one has ever retired from any of the
No.
podcasts. Some people have left. They didn't retire, though. They were asked to leave.
No,
Oh,
till death do us part, Vincent.
Well, some people they hardly show up. And I said, you don't really want to do this. So why don't you just not do it, right?
Hmm.
I'm not trying to be mean or anything, but I want people who are into it. And you guys are for sure. The whole
We
Twitter,
are into it.
everybody on all the pods right now are into it. So that's good. Okay.
Yes.
If you like what we do on these shows, or these podcasts, these science podcasts, we'd love your support to continue them. I think it's an important part of the educational world that you hear scientists talking about science. So go over to microbe.tv/contribute. Microbe TV, the production company, is a 501(c)(3). It's a nonprofit. So in the U.S., your contributions are federal tax deductible.
So you can give a boatload of money,
You could,
okay,
and you would
and
deduct it. So if you have $100 million income,
There you go.
you could give $100 million and you wouldn't pay any taxes. I don't think it works. I don't think it's
No.
1-to-1, but
A couple
Please,
of million here and there
millionaires.
wouldn't hurt, though.
Yeah.
Come on.
it's a drop in the bucket. And this is really a good cause. And we don't do anything weird here. We don't do it. We don't have any illegal activities. We don't do any
No, the viruses
laundering.
take care of that.
Yeah, that's right.
Yeah, the
Good
viruses
point.
do the weird
The
stuff.
wildlife behaves, and we
just-
interpret.
We just talk about it. You can come here to the incubator and you can see it's just a recording studio. We don't cook the books or anything like that.
No.
We're a straightforward group of scientists.
Nor
so
do we
we'd
have
love
a
your support.
political agenda.
No.
So we don't have a radical listener group.
Well, some people would disagree with that.
Some
They would
people
say
would
you,
disagree.
Well,
you do,
let
but
them.
we have a scientific agenda. And if your politics intersects with it, we're going to criticize you,
That's,
right?
That's true.
Okay. ASV 2024 registration abstract submission are now live. I would say are now infectious because viruses are not alive.
That's a
In the
debate for
meeting.
another day.
ASV 2024 is going to be in Columbus, Ohio, June 24th through 28th, 2024. The abstract deadline, February 1. If you're a student postdoc or a teacher of undergraduate virology and you want to apply for a travel award, you can start working on that applications. Students and postdocs need to be ASV members. And now would be a really good time to submit your membership application before the rush. Go to asv.org and you will find links to the membership page, travel awards page, and the 2024 meeting page. I just renewed my membership.
I was just going to ask if you had done it yet.
I did it a few days ago, and I found out I was actually 2 years in arrears.
Oh, no.
So
Made an
I had to
honest
pay-
man out of you.
I had to pay $300, and
Ouch.
now I'm going to pay another $1,700, and I'm going to have lifetime membership.
There you go.
So I don't
Oh,
have
there
to
you go.
worry about it
Yeah.
anymore.
So, and I just want to put in a good word for Kathy and her bunch here and say, dude, do your membership now. It's going to be the same amount of work whether you do it now or whether you do it later on. But if you do it later on, it's more work for them, okay? Because it gets crazy and everybody doing it at the last minute. So be a good citizen. Do it now.
I want to remind you there's still an opening for a research assistant in Amy Rosenfeld's lab at the FDA. Her laboratory works on enteroviruses. They're interested in many questions about enteroviruses. What's an enterovirus? Well, the name is not telling you. It's not just viruses that infect your gut, because rhinoviruses are enteroviruses too. Hey, that's classification, but these are like poliovirus, rhinoviruses, enterovirus 68. And a major discovery of the laboratory is that antibodies cross-react among all these enteroviruses. So what are the implications for that? That's one of the questions you would be working on. All BSL-2 works, so you don't have to worry about training. And the requirements and more information will be on a PDF on microbe.tv/TWiV, and you have an email there. You can email Amy and find out more. And don't forget to go buy Dixon's book, The New City, at despommier.com.
It's also
Not
on
because
Amazon.
Yeah, so despommier.com
That's
is
right.
simply a landing page
That's right.
which has a little bio of Dixon, a picture of him, and links to where you can buy the book. It's
Right.
just one place where you can go. I mean, you could go to Amazon, you can go to Barnes & Noble, all the other booksellers you're used
Sure.
to,
Or Columbia University Press. You can actually go to where it was
to their
first
website
published.
as well.
They will
Yeah.
send you a book.
I think I have a copy. I have a copy, so I can show
By the
you
way, this is an
cover.
unsolicited, unpaid for.
Yeah, it's unsolicited. I like it, and it's also it's autographed by Dixon. See, it's
Whoa.
autographed.
He could.
And it says here to Vincent, many thanks for all you have done to make my book sing.
That's right.
Let's
How
see
nice.
if it's in key.
Let's hope so.
B
All
flat.
right, so a couple of interesting news items in the news. We're gonna have a new section here on TWiV.
We have a
In the
lot of
news.
it today, and all of it is very interesting.
So the first one's in the New York Times, and I don't even have access to that. Holy crap! I
do.
can't read it.
I
So someone
do.
someone summarize it, please.
This
Long
is well, the first paragraph says the whole thing. A Long Island midwife falsified vaccine records for some 1,500 school-age children, according to New York State's Department of Health, which on Wednesday announced that it had fined her $300,000. So this is a woman who she's a midwife, and she gave children some sort of pellet, it's described, a homeopathic treatment. I won't call it a medication.
Right.
They were
A
given orally,
homeopathic treatment that was touted as being equivalent to a vaccine instead of giving MMR and a number of other vaccines.
Yeah. So
And then falsified the records. She
Oh
said
my
that
God.
she filled out records saying that these kids were now vaccinated, and they come from all over the place. A lot of them in Long Island, but all over the place as well. So we have a bunch of unvaccinated kids going to school. And
yeah.
she-
So it mentions in the article that there was a change in New York State's exemption requirements for vaccines, and so there were suddenly about 25,000 kids who needed to get vaccinated who hadn't before. These were some of those kids, and it sounded like at least some of the parents were aware that the kids were getting this homeopathic treatment instead of getting actually vaccinated. And the very end of the article, I found especially infuriating because it mentions that she particularly has a lot of issues with the idea of vaccinating pregnant women. She thinks that that is particularly awful. And she says something to the effect of, well, doctors don't know best.
yeah, that
about
was,
things.
I just, such an ironic statement at the end. She says, well, a doctor doesn't always know best.
She apparently didn't
parent-
go to all of her classes.
Were the parents under the impression that whatever homeopathic treatment their children were receiving was just as effective as the vaccine? Were they communicated like that?
I think that the parents may not have wanted to vaccinate their children,
Okay.
but needed to get them able to be sent to school now that they were no longer exempt.
Yeah, these were kids that the change in the laws was to get rid of a religious exemption. So there were
Okay.
a bunch of kids who had previously had fallen under that exemption.
Mm.
So,
Okay.
who knows that they don't detail the motivations in this, but it is stated in one case that, or in one place in the article, that some of the parents clearly understood, or more or less, that she was going to not really vaccinate them, at least not with a real vaccine, but would nevertheless fill out the records so that they could get into school. So the parents are, in many ways, as culpable, some of them, in this as anybody else.
She went to a very good school, and she had very good educators, and she ignored them. She blatantly ignored the information they were giving her because they obviously were in control of the information flow. They based science as their basis for recommending vaccination as a way of interrupting disease transmission and not getting sick. And she purposefully ignored that information. So that sort of, flies in the face of logic to say, well, well-educated people wouldn't think this way because they've heard, and then you can stop right there because you can shut your ears to anything that's being said, no matter where you are.
Well,
That's a shame.
this shows up all the time.
I know it does.
I'm thinking, did we already talk about the Florida Surgeon General?
Yeah.
Oh my God.
Mhm.
Yeah, we
Oh
did.
my God.
I think he has a, at least on paper, credible education as well.
He does. He has a MD, PhD from Harvard. And yeah.
Oh, you know about those Harvard PhDs.
We do.
and he's ignoring the science. Yeah. He's making
totally.
statements that are completely wrong.
That's right.
It's the same with anti-vaxxers. I'm not putting stuff in my kids. Why not? You put all kinds of things in your kids. There's nothing wrong with vaccines. Give me a break.
You can find a group that
gosh.
disagrees with every factoid.
Of course.
Like flat Earth people. I don't get it.
Oh, that reminds me. Have you seen the cartoon? It's not really. It's kind of a cartoon that shows the sun and all the planets lined up, and they're all round except the Earth, and it's flat. Okay, and the caption is something like, "Well, this is awkward."
That's very good.
Cute.
Oh, I like that. All right, another story. More vaccine-derived poliovirus circulation. Now in Indonesia, WHO is reporting 4 cases of paralysis caused by circulating vaccine-derived poliovirus type 2. And they note here that there are no hard areas to reach in the region. With some other countries, it's hard to reach. Here, they say socio-cultural barriers in East Java's Madura community pose challenges to vaccination, from vaccine hesitancy due to several reasons, including fear of adverse effects, multiple injections, and sometimes due to religious reasons. I don't understand the religious objection. Wouldn't whoever your religious leader is want you to be healthy, as opposed to making a blanket ban on say vaccination? That doesn't make any sense to me.
I think some of the religious groups have this idea that you should just hope or pray to have disease not strike you or to be protected, and that you should not do outside intervention.
Really?
Yeah, or to
Yes.
accept whatever
Well,
has been
Yeah,
bestowed upon you via the divine or whatever.
Yeah.
I think
Yeah.
one of the prime examples is Christian Science.
Yes.
Mhm.
Right. And I don't really know what somebody else, we can get some mail here and somebody can enlighten us. I forget what the rationale there is, but basically they don't want humans intervening in your, health. Okay. Let the Almighty take care of it.
It's interesting because the Pope has been ill.
Yeah.
And has been treated with traditional, that is to say, not traditional, but Western cutting-edge medicine, as has the King of England. So I think that these religious belief systems are not commonly shared, even among the people that are of that religion.
Yeah, I mean, the Pope says people should be getting
Yeah.
many of these things.
I mean, what bothers me, well, there are 2 things. First of all, young kids, they depend on their parents to protect them. And this is not doing that. So you have some religious belief, but in the end, it's not protecting your health. Okay. And the other thing is that you can transmit these infections to other people, and that's not fair. So, public health depends on everybody cooperating, but it's becoming increasingly clear that people don't want to cooperate, don't give a damn about each other. And I'm sorry to hear about that.
But
Anyway,
I mean, I think because I feel maybe it's I'm in a different news realm over here, but the Catholic faith doesn't go against vaccination or modern
No, of course
medicine.
no, it
So
doesn't.
I don't know if just in case somebody understood that we were trying to infer,
No,
we
no.
were trying to say that it's not against modern medicine at all.
No, it's
I
other.
know,
No.
yeah, exactly. Okay. I know
That's a good
that
point.
because it sounded like at one point that's what we were maybe
Yeah.
insinuating, and that can be, that could offend a lot of people or just be misinterpreted. So
Sure. We have an article from the city of Philadelphia, and this is an update on the measles outbreak. As of January 16th, they have now a new confirmed case of measles, so that brings it to 9 cases. And this is the 5th case associated with the daycare outbreak group of kids. They don't say whether it's an unvaccinated kid, but I'm guessing it is,
they
right?
must be, no? Yeah.
So, Vincent, is that an epidemic?
Yeah, I think it's an epidemic, right? What is an epidemic? That's a good
That's
question.
why I asked the question.
You know what? They call it an outbreak here in the
An
headlines.
I think
outbreak, right.
outbreak is appropriate.
So when
Because,
does it become an epidemic? How many cases do you need?
I
Let's
think
look it up.
it's dependent
Online-
upon what you've defined as the endemic level. So
Yes,
usually
that's
there's
right.
for
That's right.
For example, for flu, I know it's a particular percentage, and that's defined relative to the endemic level. So I
Okay.
don't know. I mean, with measles, the endemic level is probably so low that much of anything would be above it.
Polio, I mean, of course, and you mentioned that. And plague, I remember listening to an epidemiologist say in Colorado, one plague case is a plague case, 2 plague cases is an epidemic.
Well, it depends. Here's a definition from the Mayo Clinic. So an endemic is the amount of disease that's always present, right? The
Mhm.
baseline.
Right, sure.
And then an epidemic is an increase over the baseline.
An increase over.
So if there's 0 measles, typically, then 9 is an epidemic, I think,
Right.
right?
That's right.
Wouldn't rapid spread also be involved in the definition of epidemic, though, that it could be rapidly spread through the population in a short period of time?
It says, an increase often sudden in the number of
Mhm.
cases. Yeah. That sudden is not a quantitative word.
Yeah, it's
Yeah.
yeah.
So,
By the way, as an aside, Vincent, you said you got that from Mayo Clinic. That's kind of my go-to
yeah.
I like the
online.
epidemic too.
That and of course the usual CDC and et cetera. But Mayo Clinic is really great.
The Cleveland Clinic isn't bad either.
I also have an article from the Virginia Department of Health. They are investigating a potential measles exposure in Northern Virginia. Travel
Yeah.
through Northern Virginia. What's the possible exposure here? Does anyone know?
This was somebody, an international traveler, I think.
Yeah,
coming in.
that one was an
Mm.
international traveler. I actually read a ProMed post about measles cases in the U.S. earlier today while I was eating my lunch, and it mentioned both of these. It also mentioned one case in New Jersey, which of course I quickly went to read about, which is in Camden County, right next to Philadelphia. And the reason why I found that kind of noteworthy is that they said that they didn't that they had no evidence linking it to any of the Philadelphia cases. So part of me wonders if there might be more going on here than just these. For example, how exactly that case, presumably, which you would think might be linked to other Philadelphia cases,
The
actually has a chain.
incredible aspect to this is that if you look back 100 years from today, the outbreak rate for these diseases was incredible, even though public health had taken hold. Not the vaccine aspect to it had not, and it's such a huge contrast. How could anybody see that and say that they're against vaccination? And that's a fact. You're not even stating rumors. You're giving them numbers. We're worried about a single case of
Dixon.
measles.
They're
a
not
single case of polio.
thinking logically,
Thinking. They're not
thinking, not using their
They
brain.
don't want to agree with you no matter what.
I went to ProMed Mail because Brienne just mentioned it, and the first outbreak news is about squirrel pox in Ontario.
Oh, cool.
Squirrel pox. Wow.
Case of
That's
squirrel pox in an animal rehabilitation center in Ontario. Squirrel fibromavirus. It causes unsightly lesions on squirrels. Oh my gosh. They would not want to have their picture taken.
I'm Googling images. Oh no, it kind of
Interesting.
looks like myxoma on rabbits.
Yeah.
It looks similar.
I guess when squirrels get it, they go nuts.
Okay. Now we have, moving on, Wall Street Journal article. Title is Chinese lab mapped deadly coronavirus 2 weeks before Beijing told the world, documents show. I have a feeling that they struggled a long time to find the word that they could use that was short in the headline, and they used mapped. And you know, we use mapped in a certain context, right? You know, the real thing is determine the genome sequence, right? But it's too long for a headline, so
Yeah.
they say mapped. So anyway, what this is that I have actually the PDF here because the darn Wall Street Journal is that they had the complete sequence of SARS-CoV-2 in a lab in Beijing on December 28th, 2019, which is about 2 weeks before it was officially released. Okay. I think that's kind of crappy that they waited 2 weeks, if that's the case. Now, I just don't know if that's, the numbers say.
What are the dates we're talking about?
So December 28th, they uploaded a sequence to a China website, or U.S. government-run database, December 28th, 2019. And then they officially released the sequence on January 11th. So it's about 2 weeks. So I think that in itself is, if that's true, it's unfortunate. And a good thing is that the article, which is written by Warren Strobel, says this doesn't shed light on the debate over whether the virus emerged from an infected animal or a lab leak, which is true. It doesn't mean anything.
No.
However, I mean, 2 weeks could have been useful for the world, right?
Yeah.
But Jesse Bloom says we can't trust China anymore. And, I think that's crappy. That's what I feel.
Yeah, I totally agree. I first saw this story on CNN yesterday, and was looking at and read a CNN article about it. And a couple of the things that I kind of found interesting in the article that I had originally read is that the way that they phrase it is that the sequence was submitted to GenBank on December 28th, and was rejected because it was incomplete and didn't have all the information that they wanted.
I see.
And then it basically got resubmitted later on. And, some people were like, how dare they reject this important sequence on December 28th. But, the first ProMed Mail post about this was on December 31st.
That's what I was going to ask. You mean about the disease?
Right, about the disease. And so, in fact, on December 28th, this was some random curious sequence, as far as anybody knew. And if you have a mistake, if I posted a sequence today and it was a random sequence, people would probably reject it if I did it incorrectly. So I felt like it made the whole thing seem slightly more understandable to think about it in that way.
In fact, on December 28th, if you have the sequence, it's a coronavirus,
right,
no matter what mistakes you make,
problem,
right?
Yeah.
It's a coronavirus, but the government is still saying we have an outbreak of unknown origin, so that's not right.
True.
They should have said it's a coronavirus of some kind.
Yeah.
And so
this tells us maybe there was more information out there earlier than some of the rest of us knew about, but I'm not sure that it totally gives us other info.
Problem it does do is that it fuels the speculation of people saying, you see, they're hiding stuff, so they're obviously hiding the origin as well, right? That's just
Mhm.
nonsense. So this is why this is unfortunate.
Well, it was also unfortunate that they withheld data for 2 years from SARS.
SARS-1, yeah.
And
Yeah,
that
it's true.
means that they're suspect no matter what happens.
I don't think it was 2
Uh,
years.
Well, it
It
was
was,
long.
it was months. It was months.
Yeah, it was months. $65 billion worth of tourist trade was lost as a result of SARS-1. And
So-
that was the reason
Yeah.
why they were reluctant to release any data that suggested if you come to China, you're going to die.
Yeah.
So,
Someone in this article says, therefore, we cannot trust any of the so-called facts or data provided by the China Communist Party, which is just rhetoric, in my opinion.
yeah, but that's not the Communist Party. That's the Communist. I mean, the Chinese scientists, not the Chinese communists, right?
Well, I don't know who was regulating the release. Because
I
if
would
you
say
remember,
somebody was.
you remember there was a guy in Wuhan, Zhang, who was convinced by Eddie Holmes to release his sequence. And he got in big trouble because it wasn't approved by the CCP. Okay. And I asked Eddie on a podcast what happened, and he said, I don't want to talk about it now because something happened to the guy.
But
'Cause he
isn't that the
released
guy
it.
that they made him take it down? It was up for a few days and then they made him take it down, or is that a different instance?
That's different, I think. So this
Okay.
one went up, and then it went to virological.org, and then everybody had it. And in fact, it was the sequence that was used to make the mRNA
The vaccine.
vaccine.
Mhm.
So you could have had a 2-week jump on making a vaccine. But I think the bigger picture is if in fact they didn't release it quickly, that's not great. But I'm not sure that, as Brianne said, I'm not sure that's it.
Yeah.
All
Not
right.
necessarily like something nefarious
Yeah,
behind it. If
right.
it was just like a nomenclature, something was wrong with the name or the file or whatever.
Yeah, that part seemed like it was a nomenclature thing, though. It
Mm.
would have been nice if, they were sequencing it. They probably knew some people had disease, and they should have told somebody.
And let me just get something straight, folks, because I know you're going to write in. I'm not defending China. I'm defending Chinese scientists. Okay, I'm defending China's science because they are scientists like us, and they want to get answers. They don't want to hide things. And what the government does, that's another story. I'm not an expert on it.
Certainly in the U.S., you wouldn't see any sort of government interference with science.
Oh, yeah.
right?
Well,
Yeah, I can't think of
speaking-
any examples.
When?
Yeah,
Of what?
What?
Speaking of that, so here's our last article. Science Insider. Wisconsin bill to restrict pathogen studies worries scientists. So you may remember the U.S. House of Representatives some time ago, we talked about this, passed a bill banning gain-of-function research. It hasn't gone to the Senate yet. But here now we have the Wisconsin House, or whatever it is. I'm not sure what the thing is called, but the Wisconsin bill, the state legislature has a bill going through that will ban certain gain-of-function studies. And this is because some people think that gain-of-function research led to the creation of SARS-CoV-2, which couldn't be a more stupid conclusion to make. Okay, I will just say it out there. I've never said that before. No, I'm just kidding. I have said it before. It's just stupid to say that it resulted from gain-of-function research, because you just don't know what you're talking about if you think that that's the case. So these bills now are trickling through. I think there's one in Texas too, right, Rich?
Yeah. Matter of fact, I think the Texas one actually, I think it passed. I'm not sure. But yes.
All this
The Florida passed one. Texas
Florida
is being
passed one as well. That's right. Last year, they enacted the first state-level gain-of-function. It uses the federal definition of enhanced pathogens, so that's better than
because
some
at
of the
least it
others.
gives some definition to
Yeah.
some definition of what gain of function is.
Because I think a
this
lot
is
of it
this-
is just misunderstanding of the general public. They also get confused as to what gain of function means,
course.
and they think that it's only this like terrible thing that you're creating some super virus that's going to kill us all. That's what
Right.
everybody thinks when you talk
That's
about
right.
gain of function.
That's
with
right.
The lay public.
So during these state hearings, do they actually have the right number and kind of people
No.
giving testimony.
They have probably all the people who are against it,
Yeah.
like
Mhm.
Richard Ebright, who's quoted in the previous article about China, and all the naysayers who say this is terribly dangerous and we shouldn't do it. They don't have any people who say this is what we all do and
And
it's
in fact,
safe.
this is another example of where we have well-educated, experienced scientists who are spouting nonsense.
Yeah, that'd be a good way to put it.
Comes out of the back end of a bowl.
I also want to, sometimes want to look at them and say, you guys get that you have the University of Wisconsin-Madison that's, a really preeminent virology research organization that,
Yeah.
is it a thing that's sort of a big positive for your state, and now you're handcuffing them? That's not a good idea.
No, not well thought out.
But the other thing that's puzzling is that Madison, Wisconsin happens to be the capital of Wisconsin also. And the Capitol building is walkable from the university campus. And it would absolutely stun me to realize that there was no open call for opinions, but they might have said, well, it's between 2 and 3 o'clock in the morning on Sunday or something, just to make it impossible. But I think there's enough scientists at the University of Wisconsin to
Yeah, of
counteract
course.
all of these arguments.
All
I don't
this,
know why
all
they
this,
haven't done it.
all this gain of function nonsense began when Fukuoka and Fouchier separately adapted avian H5N1 to transmit through the air among ferrets, and then everyone went crazy because that's a gain of function experiment.
But I wonder, I was thinking about this. I wonder, we probably discussed this before, whether that would have blown up the way it did had it not been for the reporting on Fouchier's talk at a conference where he apparently spoke about how he had created this dangerous virus, right?
Yep. I don't
know. So, it has as much,
to know.
it may have as much to do with publicity as anything else, okay? And now it's got a life of its own. Gain of function is a dirty word.
And I think that for the listeners, so the American Society of Microbiology, if you Google, they have a very simple, nice definition of gain of function. It says, what is gain of function research? Gain of function is a broad term that can encompass almost any type of research aimed at understanding mechanisms and processes. Gain of function techniques are used in research to alter the function of any organism in such a way that it is able to do something it couldn't before. That's it. It's like, but then somehow this is applied to every virus is going to kill us via gain of function research. But gain of function is not, like, the negative connotation associated with it is so unfortunate because that's literally what so many of us do in the lab all the time. And we're not being labeled as, I guess, like mad scientists for doing that.
It's kind of sad, though, that
it's
we've
kind of
got a rich
necessary.
history. Humans have a rich history of using infectious diseases as a weapon.
That's
And
true.
whether that was gain of function or not, that you could argue one way or the other. But we fear, I think innately fear, that some super infectious agent will be released that they have the vaccine for and we don't. And the next thing you know, we don't exist.
They should really check out what nature's doing in allowing some of these viruses to mutate.
Oh, that.
If they
I
think
thought you meant
those
the
ideas are
journal.
scary,
Yeah.
they should
Nature,
see what just random mutation can
does it
do.
already and better.
No. SARS-CoV-2 was a product of nature, for no question. I mean,
That's right.
the market just enhanced its ability to find humans, but product of nature for sure. And it's going on every day. In fact, one of our papers is going to address it.
Vincent, did we do the vaccine-derived poliovirus article?
Yeah.
Was I
Yeah,
asleep?
we did.
Okay, I was asleep.
Yeah. Well, we didn't spend a lot of time on it. So I should
That was
actually.
where we started talking about religion.
Yeah.
That's it.
So a couple of cases. The point of that report is, okay, a couple of cases of vaccine-derived polio in Indonesia is that it's very hard to eradicate poliomyelitis, and it's impossible to eradicate poliovirus. But the attenuated vaccine is very problematic, and the development of NOPV2 has not fixed the problem. And so what really disturbs me is that people at WHO and the Gates Foundation, who's funding a lot of this, they're just blindly gung-ho saying, oh, we can do it if we just push. No, there's a biological problem that cannot be overcome by pushing, folks. We need to have another approach, and you got to stop shutting down polio research because poliovirus is everywhere. So it makes no sense to keep it out of the laboratories. Let's make some better vaccines. Otherwise, we're never going to get rid of the cases of polio. And this has been going on since 1980, folks. How many years is that? 20, 43.
40 something.
44.
44.
Before I was born. No idea.
Ooh.
I'm
Really?
joking. Sorry, I had to say that.
Is it before you were born?
Of course.
Oh, yeah, you're right. 44. Holy shit. Yeah, this is not working. Rich, if you did an experiment for 44 years and it didn't work, you wouldn't have gone that long, would you?
No.
Yeah,
No.
none of us would.
43 maybe. 44 no.
Nah,
Okay,
a
okay.
PhD is long enough, 5 to 7 years, and then
Anyway. That's the whole point of that. It's
Mhm.
just, and we get more and more reports of vaccine-derived cases. It's not the right approach, folks. But however, we have two cool papers for you today. All right, the first, PNAS, Proceedings of the National Academy of Sciences. This is an article called A Plant Virus Manipulates the Long-Winged Morph of Insect Vectors. Now, a virus does not manipulate anything. Okay, it's the wrong word, but I won't get into it. I just want to focus on the science.
I actually mentioned in my class that I knew someone who really, really didn't like anthropomorphism, and we should try not to do it in class.
I don't like anthropomorphism, but I do like wing dimorphism,
That's right.
right? Where you have an insect and some forms have no wings or little wings and some have big wings, which is very cool, right?
You bet.
So this is very well known, and it's thought to be an adaptation for changing environments, right? And, you know, when you have a bigger wing,
You can go further.
you can go further.
That's
Have
right.
wing will travel.
You got it. You can wing it, so to speak.
You can wing it. So you can go to
But
new
there's a-
habitats. And
But
of course,
there's a trade-off, right?
of course. What's the trade-off, Rich?
Trade-off is lower fecundity.
That's
You don't
right.
reproduce
especially
as
in
well.
females.
Especially
You
in
gotta
females.
put more
Especially in
resources
females.
towards making the wings, and you can't put those resources towards
Yeah.
making
There's always
a progeny.
there's always a trade-off. You know, there's always a
Right, a
trade-off.
loss of function.
And so,
Yeah.
if these insects have viruses, obviously, making big wings will help transmit the virus right to different places. And so,
It sounds purposeful, doesn't it?
it is evolutionarily selected, Dixon. That's how I would put it. I don't
Yes, I
think
know,
you
but
can
some
say
people
purposeful.
say
No, it's not
this is a
purposeful.
wonderful expression of God's will.
All right. That's a bit much. So the well-known way to have wing plasticity is by changing the physiology of the insect, right? There's a pathway in the insect that can change from no wing to having a wing, and it
That's right.
involves the insulin-like growth factor signaling pathway and other factors involved with it. So that can change. So that's obviously responding to some environmental conditions, and it can change how the wings are developing. But in this paper, we're looking at how a virus can affect wing development, right? And we are looking at plant hoppers, okay, which are vectors of viruses. So plant hoppers can go from plant to plant. There's the small brown plant hopper, which is the topic of this paper, and then there are brown plant hoppers and white-backed plant hoppers, and these are crop pests as well, and they transmit viruses and so forth. And one of the viruses is rice stripe virus, one of the most economically serious rice viruses in East Asia. And this virus is a TWiV virus.
Did we say that this, who the authors were and where this comes from?
I didn't. I
We
just got
didn't.
so excited.
Yeah. This is from the Key State Laboratory of Integrated Management of Pests and Insects and Rodents, blah, 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 in Beijing. Okay? So rice is important in China. So you can imagine there's a motivation to focus on this. One,
Well,
two,
yeah.
3, 4, 5, 6, 7, 8, 9 authors. Are some of these, I don't know what the...
And
I don't
reading
think
through
there's
this
some of
paper,
them-
the rice stripe virus, RSV, I couldn't stop thinking about it. It
Yes,
was so difficult to not
absolutely.
think about like
Rouse
respiratory syncytial
or respiratory,
virus.
yeah.
So we
The
have
whole
two senior authors, Kang and Ki, and we have two equally contributing first authors, and those are Yu and Zhao, with Chen, Lu, Zhao, Li, Lu in between. Okay.
So this is a virus that infects plants. So the leafhoppers pick it up. They get persistently infected, and they can transmit it to other plants and to their offspring as well. It doesn't hurt the leafhopper. It's a good evolutionary development, right?
So they didn't actually, I don't recall that they actually addressed this specifically in the paper, but this virus is actually replicating in the insects as well as as well as the plant. Is that correct?
I believe I saw a sentence about it replicating in the vector. Yeah.
So
But they
the
didn't
other
say where. I
Didn't
also
say
thought
where.
they didn't say if it was in the wings or where or
Yeah. Actually, it's in testes, right?
Oh, they did talk
Ah,
about
right.
testes actually.
Which
That's
is
true.
really
And
strange.
the ovary. That's true. They did.
So
But,
I looked up, I looked up the virus, as I always do, because I wasn't familiar with this dude, and it's a segmented virus with 4 single-stranded RNA segments that are
Right.
either negative. They're circular, and they're either negative sense or ambisense. That is, both senses are used. And the weird thing to me is that, as far as I can tell, it's not actually encapsulated. It's in the traditional sense. Okay, the segments get wrapped in a nucleocapsid, all right, and transported from tissue to tissue in the plant at least, and I think through in the insect as well. But you never see 1 or 4 segments in what I would regard as a typical capsid.
So that's kind of like tobacco mosaic virus, where it's just a protein bound to the RNA, right?
Mm-hmm.
So you could call it, it's not really a nucleic capsid protein because it's not a substructure, but it seems like it's the same thing.
Right. So it's fair to say, though, that the major disease of rice is caused by a fungus called rice blast. And that's what really wipes out the crops every year. It's an airborne spore. It's it's impossible for the farmers to do anything about it. When it gets in the air, it just decimates. It does worse than that. It eliminates the crops. Leafhoppers with this virus, I don't know what the rate of crop failure is, but it's got to be a lot lower than rice blast.
All we know is that it is one of the most economically serious rice viruses in East Asia.
Well, rice viruses, yes.
Yeah, is what they tell us.
I could never disagree with that.
So it is known that viruses can affect wing development, but they don't know the mechanism, and that's what they're trying to figure out here. So this virus, RSV, rice stripe virus.
Yeah, not the respiratory RSV.
Not respiratory, can induce a long wing morph in male SBPHs. Okay.
And they never defined, like they never actually wrote, unless I missed it, within the text, the acronym. Like, of course, it's like
they did.
Yeah,
Actually,
they did.
I looked
Short
it up.
body plant hopper.
Yeah, so that I inferred, but I didn't actually see it. Okay.
Or no, there it is. Yeah, it's
Paragraph 3.
actually it's small brown plant hopper, and it's
Oh, small.
Small brown
and it
plant
is
hopper. Yeah.
it's defined right there.
So the virus can do this without these environmental cues, which is the other pathway driven by the insulin-like growth factor. Okay, and so what they do is they have these plant hoppers in the laboratory, and they see that the adults have. So it's the adult that develops the big wings, and then in the non-winged insects, they're just buds, little buds there where the wing would be. And so they compare the wing dimorphism between insects with and without the virus. And I like the use of the word viruliferous.
I thought that was amazing. I've never even heard that word used like
I
that
think
before.
we've encountered it before. It's a great
And
word.
non-viruliferous.
Yes. So with virus and without virus, right? And they found that if the long wings are in 79% of the insects with long wings have the virus, and only 37% of the long winged don't have virus in them. And of course, they can still have long wings. Those are induced by the other pathway, so that's okay. But all the females only have short wings. So it's a male specific thing. Even if the females are infected with RSV, they don't get long wings. And they can do an infection experiment. They can make an extract they can grind up the infected insects and then give it to other insects, and they will increase the long wing development from 39 to 72%. Okay, so, and they can also, this is an interesting experiment. They also can knock down the virus with double-stranded RNA, and it doesn't matter. You still get long wings. And they conclude that you don't need a lot of virus to do this because the knockdown is not 100%.
Yeah, they said it was independent of the titer in all
Yeah.
circumstances, in all cases. No, so just a little bit of virus will give them long wings.
So this is in the laboratory. So what about in the field? Okay, so that's a good thing, right? Is this work in the field? So they go out and they catch SBPHs from 9 field places in China from June to August. And the males were all 100% long-winged morphs, and over 70% of them had virus, and the viral titers varied greatly. Okay, so those ratios agree with what they found in the laboratory. Now, this is an interesting. So they did this knockdown experiment with double-stranded RNA. I just mentioned that to try and reduce the viral titers and see the effect on making big wings. And the control for that was a green fluorescent protein RNA injection. And this is the beauty of science. Sometimes things happen that you don't expect. They noticed that even if they put the control RNA in, you still reduce the amount of virus. You reduce the amount of wings. Sorry, not virus. The amount of wings, long wings that were made. And so they hunted for that. And it turns out that the sequence of GFP is very similar to some genes in the insect.
Kidding, of course. Well, where does GFP come from?
From
An invertebrate.
That's right.
Yeah, it comes from
a
fireflies, right?
fireflies. That's
No,
right.
so that's
No,
from
it's
jellyfish.
jellyfish.
That's
It's jellyfish.
That's
I'm thinking
luciferase.
luciferase, yeah.
Okay.
Hmm.
This from
Jellyfish.
jellyfish. You're thinking of luciferase.
I am.
right?
I'm sorry. Okay.
But
Take my question back.
anyway, so there's a gene that they call contig 2490 that is the one that decreases the long wing ratio. And they say, due to the unusual detection process, we name contig 2490 the encounter gene.
I love that.
It's
Encounter.
cool. I really like that.
Yeah.
So now it's encounter. It's encounter. And it has so this is a protein encoded by the gene, and part of the protein looks like a malate dehydrogenase, which is usually in the mitochondria, but it doesn't have any such activity. So it's a plant hopper specific gene, but it doesn't have any malate dehydrogenase activity, but it can somehow be involved in making long wings. So how does this work? They look at the expression of encounter, and they find in the males it's predominantly expressed in the testes. So I spoke incorrectly before. I said the virus reproduces in the testes, but it's encounter that's in the testes. Okay.
Mm.
I'm not sure where the virus is reproducing. So encounter is mostly in the testes. And in fact, it
And
has to be.
ovaries. No? Oh, no, there was none in the ovaries. Sorry, there was only a little bit
Yeah.
because they have the ovary as well.
But
there's-
interestingly,
Hmm.
it is so the development of larvae goes through instar stages, right? First, second, third, fourth, and it has to be it's expressed early. It seems to regulate wing development before the fourth instar larval stage. Dixon knows all about instar. Probably also Brianne does too, because you're in a biology department, right?
A little bit. But Dixon, I am certain, knows more.
Dixon, what's an instar stage?
Because I'm
Dixon?
older than you. An instar is just the name for that particular developmental stage. And you were right. There are 4 larval instars and an adult So the 5 altogether, 5 stages
Right.
to complete metamorphosis. That is a complete metamorphosis.
Are instars demarcated by a molting?
Molting.
They
yeah.
are. They are.
Okay.
And that's true from nematodes all the way up through insects. Now,
So-
what I'm
so
wondering
the-
about when I heard about this paper, by the way, this is not an open access paper, so I can't actually see a lot of the stuff that I'm hearing about right now. But there are so many insects that have developmental wing development at a specific time.
Yeah.
When, for instance, when ant colonies and termite colonies get overcrowded, they suddenly leave. And how are they going to leave? They leave by flying. So they become an alate stage. They grow wings and off they go. Now, that may not be a viral induced affair, but so do locusts.
Mhm.
Locusts remain as tiny little grasshopper-ettes until the conditions turn just right. Maybe the odor of a crop wafts over the field of these
Yeah,
immature,
could be.
and the next thing you know, they've got wings and they're flying towards the source of that odor. So I wonder if viruses play a role in that as well.
They
Maybe.
do. They do. We've actually
do.
talked about viruses that aphids pick up. They deliver to plants. The plants make volatiles that attract more aphids, and they feed on them and they get infected.
Well, that's cool.
But Dixon, I want a virus that'll make me have wings.
They do point out, Vincent, that encounter was not found in other insects with
I
the
know.
search in GenBank. So
I want
maybe
wings.
if
He wants
encounter
wings.
was in
Wouldn't it
some
be
of those,
cool?
they get
It'd be
wings.
so cool.
Yeah. Oh,
I wouldn't have to take the frickin' train every day.
I know your commute would be
Listen.
so much easier.
There's a wonderful song that is, if I had wings of an angel, I would fly over the prison walls. And it's kind of poetic to think about being trapped somewhere.
Going
because you
into
can't
the
fly.
prison or out, Dixon?
Brienne, if I inject
Yes.
myself with encounter protein, do you think that would work?
I don't think it would work. I think if nothing else, you'd certainly need some rice stripe virus to be involved too.
Okay.
So, so is this specific for these small subsets of leafhoppers, or is it true for all leafhoppers?
I don't think they just say it's
They
the
looked
Well,
at-
gene.
know that.
Yeah, they looked at 3 leafhoppers.
Yeah, so
Right.
they say it's plant hopper specific because it's not found in others, but how widespread, I don't know.
I mean, plant hoppers, they are one of the most diverse groups of insects.
Mm-hmm.
They're in a subclass called Erythroneura, and they're beautiful animals. When you get up close to them, they're absolutely spectacular in their morphologies. They've got all kinds of lumps and bumps and they're almost invisible. I mean, they're less than 2 or 3 millimeters long for the most part, and they're one of nature's most exquisite evolutionary products, shall we say.
So one of the things I neglected to tell you is that they also looked at the expression of the levels of encounter, and it's increased in viroliferous insects. All right, and it goes up first, second, and third instar. So, and then in non-viroliferous insects, it doesn't go up. So somehow the virus is inducing this, and that produces the long wings.
Yeah. And Dixon also, like I said, they say that it isn't found in other insects in GenBank. And my first question when I read that sentence was, what fraction of insects do we have in GenBank? And so I'm not sure we
Exactly.
can really answer your question because I don't know how many insects have been sequenced and added to GenBank.
My guess is you've got all the ones that impact our food supply.
For sure.
and
Yeah.
vector transmission.
And
also, of
vectors,
course.
yeah.
So they want to know how this works. So they look at mRNA levels, transcript levels in the insulin-like growth factor pathway, which does the physiological induction of long wings, and they find that encounter doesn't work through that pathway. Okay, it's got another pathway which is called the AKT pathway. It's downstream of that. And infection appears to upregulate AKT, which is a very broadly acting transcriptional regulator in many different organisms. And so somehow the infection is upregulating AKT, and then it somehow leads to the induction of encounter, which then in turn makes the big wings. So how does a virus do this, right? They make small RNAs to knock down viral specific RNAs to try and understand what's going on. And they find they have viral specific RNAs which can activate or inhibit the transcript level of encounter, and they identify a specific activator and a specific inhibitor that affect the levels of encounter. So from the viral genome is made small RNAs that can activate encounter, and there are also some small RNAs that can inhibit encounter as well. So there's some regulation going on there, which is quite interesting. So if a virus infects cells, small RNAs are made by degrading the viral RNA. It's part of a defense mechanism. Those RNAs can activate encounter and the transcription, in fact, of encounter. So that's pretty cool. So that's the story here. We have a virus that can infect males and induce long wings, and presumably that's an evolutionarily selected property so that the virus is spread far and wide. And as we said before, females don't have this response to infection because there's a cost. It's an energy cost, right? And they need to put their energy into egg production. So there's a compromise between flight capability and fecundity. I see a lot of women pilots these days, so it doesn't apply to humans. Do you know that women are better pilots?
Doesn't surprise me.
I read somewhere.
I'm joking.
Because all these Emirates and those kind of airlines in the Middle East, all the pilots are all women. So I read an article and they said they have better flight responses than men do.
The recent Boeing incident, where the window blew out, that was a female pilot who apparently handled it really, really well, according to the
I think that's the thing, that they can be readily trained to respond beautifully, and that's why. Well, anyway, that sentence, flight, a compromise between flight capability and fecundity, is very interesting. All right, so that's how a virus can help you grow wings, and despite what Brienne said, I want to get some encounter.
You just have to get some rice stripe virus too.
Just get some RSV, the new kind of RSV. Go see.
RSV number 3.
So there are many examples of how viruses have these interesting effects on their host. Wing development is one. There are many others. There's one where the virus infects an ant and makes the ant climb to the top of the tree canopy where it normally doesn't go, and then it gets eaten by birds who then bring the virus far and wide. Isn't that cool?
Crazy stuff.
And then there's the other one that infects caterpillars. They crawl to the top of the trees and explode and spread the virus all the way down the tree.
Is that virus or is that fungal?
That's virus, the one where the caterpillars explode.
There's a fungus that's similar.
The fungus is a fungus that makes something grow out of the head of the ant.
That's right.
So cordyceps.
I have a correction to make.
That is
Erythroneurons and leafhoppers have an incomplete metamorphosis. There's an egg stage, and when the egg hatches, it looks just like the adult, except that it's much smaller. And then it undergoes 4 molts to become an adult, as opposed to those that pupate and then hatch from their chrysalis. Okay.
All right.
My error.
Right. Our next paper, PLOS pathogens, reverse zoonoses. All right, let's stop. Reverse zoonosis. A zoonosis is an infection that goes from a non-human animal to a human. And zoo is the same origin as zoo. You go to the zoo to see animals. Zoonosis. A reverse zoonosis goes from humans to non-human animals. Got it?
Yeah,
Also
there
known
ought to
as
be a different. There ought to be a whole new word for that.
Well, there
Well,
is
there is-
zooanthropanosis.
Is zoonopsis, right?
Exactly.
But yeah, people don't use it. Well, some people do.
It's a long word.
Reverse zoonoses of 2009 H1N1 pandemic influenza A viruses and evolution in the U.S. swine results in viruses with zoonotic potential. I picked this for you, Angela, because pigs are involved. I
I
know
like pigs.
they're not as cool as cows, right? In your opinion?
No, but they're better than chickens.
Wow,
So,
I'm
Angela,
joking.
this is really something.
Pigs are so smart, though. Pigs are extremely intelligent. They're actually one of the top 10 smartest animals, which a lot of people don't know, with primates.
Yep.
Which is why, like, their intensive farming and the lack of like well-being and regulation around their well-being is so sad because they're extremely intelligent.
What are the other 9 top 10?
I mean, there's like all of the great apes and then other primates.
Are
other
octopi
monkeys.
in there too?
No. I know
They're
people think
not.
octopus are really smart, but they're not. No. But
What about
elephants are.
dolphins?
I think dolphins are. So dolphins are
Definitely.
actually, by our definition of intelligence, which is brain mass to body mass, when you compare the ratio, bottlenose dolphins are actually the smartest species and technically smarter than us by our own definition.
Right.
So
I found an article, and they had the 30 most intelligent animals in the world, and they have a picture of pigs at the top. So we got humans number 1, dolphins 2, ravens are number 3.
Ravens.
Oh,
Wolf
wow.
the raven, nevermore.
That's right.
Pigs are number 4, chimps are 5, elephants are 6, African gray parrots are 7, octopuses
yes.
are 8. Hey, Angela,
okay, so they made top
octopus-
10. They made the top 10. Okay.
bonobos are 9, and rats are 10. Oh my gosh!
Wow.
Then he goes on.
But pigs,
so wait a
pigs.
minute,
So many people think that they're not intelligent animals, and we have them like
Oh, no,
farmed
no,
intensely,
no.
and they're
Yeah.
so smart, which is why, like, being certain enclosures that are pigs are not in the best conditions and in intensive farming. It's really sad because they're extremely aware of their surroundings and they're very smart. So like environmental enrichment is very important for swine, but.
Angela,
Yeah.
I probably missed an episode here. I want to know why in your life cows are cooler than pigs.
Oh no, I
Do you
just
want to know
really
about the
like
cows,
cows.
or do you want to know about the chickens?
Yeah, the chickens are just like, the chickens, we talked about Merrick's and I was just joking about how I liked cows more than chickens. But because cows are just, like when I was in vet school, I love cows. They're docile. They're very like nice to be around. They're most of the time easy to treat. Like compared to horses, like we were talking about the rectal exam, like Vincent and I have for a few episodes now. And and with horses, it's very dangerous. You can still get kicked by a cow, but a horse is like it's like way more dangerous and the chances are a lot higher. But cows are just so beautiful. Like if you've been around them, they're just so like calming, and I don't know how to explain it. But
Okay.
I also really like pigs. Pigs are nice, but pigs are a little bit more, they have more like attitude because they're really smart. So you
So
have to.
Johnny Carson's late night television show, at one point, someone raised the question as to whether pigs or horses were smarter than the other, and that was
Pigs.
an ongoing argument for about 10 shows. They
Hands
finally
down.
got experts on horses and pigs together on the same show, and pigs by a long shot were ruled
Huge.
the
'Cause, like,
the superior
are
of the
just-
two.
Horses are also just like they're prey and they're extremely like their fight or flight response is so strong. They're easily spooked. They're very nervous. They're
Yeah,
very
that's right,
timid.
that's
Like
right.
it's very hard to have a, I guess, good relationship with a horse if they're not your horse. If they're your horse, yes, but even so, they can get spooked very easily, and they can even injure themselves because they go on these like adrenaline rushes, and yeah,
If not
Lots
for
of-
humans, they wouldn't exist.
true.
But
Yes,
um,
evolutionarily they don't make sense. These like monogastric herbivores on like skinny legs, and they're so heavy on top, and they have all this extra space in their abdomen, so their intestines get all twisted and they get colic. It makes no sense evolutionarily.
this is
Like,
great. I love
It's
it.
just, like, and their cecum is like floating around. That's most of the time, like the cecum flips on itself, which is why horses get colic because there's so much space in their abdomen. Like, how does this make sense? Why would they be running so fast that this can just like flip? Yeah, evolution
Wow,
sometimes.
what a rap.
That's a good rap. I love it. That's a TikTok. Angela, you said pigs have an attitude. So how does that manifest?
Well, I mean, they're very aware of their surroundings, right? Like when I've gone to pig farms and you have the sows in like these extremely tight cages where it's not
Yeah.
even a cage, it's like an enclosure where all they can do is lay down and stand up and the piglets kind of run around them on each side. And obviously the sows aren't in like they're not happy. They're not, I don't want to say in a good mood because it's making it seem like it's like a human, but they just don't seem like they're in the best conditions and they're aware of
Yeah.
their conditions. Like when I've gone into pig, I actually don't consume pork. Most people think it's because I'm like halal or something or like kosher, but I actually don't consume pork because I've been so traumatized by every swine farm that I've been to.
Wow.
And the look in a pig's eye, there's just something, I don't know how to explain it, but like when I've gone to farms, like the look that they give you is they're very aware of their surroundings. And I've just seen like this suffering that I don't see in other animals. I'm not saying that
Wow.
other animals don't suffer, but they're just extremely aware.
Yeah.
And like when they're in the slaughterhouse, they're aware. They're more than like other animals. And I know that's like a touchy topic, but it's just like, yeah, it's just from my own experience.
So, Angela, the way they husband pigs for production
Mhm.
relates to the fact, and you know this, so I think you should be saying this, but I was told that, and because remember, I did my research on Trichinella spiralis, and it was transmitted to people from pigs.
Yeah.
So the pig husbandry is a big subject. So these cages are designed to prevent the mother from rolling over on her babies. And in a free range, so to speak, piggery, a lot of babies die because they try to get to the teat first, and the mother wants to supply everybody with food, so she's very magnanimous, and so she rolls over to make room, and there's poor little Louis that just got crushed.
Yeah, it's true. But then at the same
So
time, if it means like increased well-being for the mother and for the rest of the piglets, so the mother can actually interact with her piglets in a positive way. And if one or
that's
two
right, that's right.
piglets die, because most of the litters are 15, 20 piglets. There's so many of them. And there's
That's right.
always runts. You always
That's
see runts, and they always unfortunately end up dying. And in the way that they're enclosed now,
Yeah,
you'd walk by and you'd see dead piglets all the time, just like sitting in the corner or piglets that
that's
were
right,
dying.
that's
It's
right, that's
horrible.
right, that's right,
Like,
that's right.
and then they're removed once they're cold and like already like through rigor mortis, like the next morning, like, oh, this piglet.
Yeah.
I'm sorry if this is graphic for the listeners. I'm sorry, but like it's just
No, it's okay.
it's just a reality. It's
Interesting,
a
yeah.
reality.
What you said, the look in their eye,
It's
they're looking
yeah,
at you and saying, "Get me the hell out of here."
I
exactly.
presume
And
you didn't get bored by working with pigs.
definitely not. I love pigs.
No,
I
no,
love
no,
pigs.
Angela, that was a joke.
That was a pun.
Oh,
Yeah.
boar,
Dixon
like
is on.
wild boar. Okay, so sorry.
So a female pig is a sow? Is that right?
Sow. Yeah.
Yes.
And what's the male pig?
A
I think
boar?
is it also
A
just
boar?
a boar? Yeah, just
A
a boar.
boar?
Like
It's boring. So
a
cow
man.
is the female animal, right?
and a bull is the
And the bull is
Yeah.
the male.
Mhm.
Okay.
You learn so much on this show.
Yeah, I mean, someone said to me the other day, "I like cow meat," and I thought, no, you're probably not just cow that you're eating. You're mostly eating
Steer.
steer, right?
Steer.
That's right.
Mhm.
Okay. Anyway, this is about pigs and influenza viruses, and these are farmed pigs, not feral pigs. These are because we have huge pig farms throughout the world, and it's a problem for influenza virus because they're infected with many different strains of influenza virus. It goes swine to human and human to swine. So there's a big problem, and this paper starts to look at that. There are a lot of different isolates. So in pigs, U.S. swine, there's H1N1, H1N2, and H3N2 subtypes of influenza virus. And in each one, they're mostly derived from human influenza virus at some time in the past. So they get infected. Humans pass the virus to pigs. You may say, how does that happen? Well, people who take care of pigs,
Mhm.
they bring the pigs to fairs and people kiss them and then they put them back and they transmit. So viruses go from humans into pigs. Then they evolve and they reassort with other viruses and they have potential to go back and infect humans. And so that's what this paper is all about. And so you may remember in 2009, the first pandemic of 2000s was an H1N1 swine origin influenza virus. That virus originally infected pigs in 1918. So you remember the 1918 pandemic infected humans. It also went into pigs for the first time. And that has stayed in pigs ever since and has slowly evolved, that virus, and then reemerged in 2009. It reassorted with a lot of other viruses, but it reemerged to cause the 2009 pandemic. So originally derived from the 1918 virus.
90 years later, yeah.
90 years later. So this is a virus they call PDM09, Pandemic 09. It has genes, the influenza virus has genes from influenza viruses of birds, humans, and pigs. And probably was in pigs for 5 to 10 years before it got into people and caused the pandemic.
And if those that are wondering from birds, it's because also influenza viruses that infect birds can also infect pigs. And if they're free range outside, they're in contact with waterfowl and things like that, so they can easily get infected.
So, Vincent, isn't there another virus called swine flu, which has nothing to do with this virus?
Well, swine flu is just a generic name for influenza viruses infecting pigs,
I'm
right?
sorry.
It's the same, yeah.
So the 2009, a lot of people call it the swine flu pandemic. Well, it originated in pigs, but once it's spreading in humans, it's no longer a swine virus. So that's not really PDM09 is the best name, Pandemic 09 virus, but
it's true that the general public does say like swine flu. They're starting to say H1N1 now. For the longest time, though, they did say swine flu.
They did.
So in this paper, they look at sequences of the virus isolated from humans and swine between 2009, when we first had the pandemic, and 2021, and try and establish phylogenetic relationships and say what came from who, right?
Just to go back for a second, I don't think we said the authors or...
Oh, true.
Gosh. We didn't. Go ahead. Sorry.
Here either. So the first author is Alexei Markin. The last author is Tavis Anderson, and these authors are from the National Animal Disease Center at the U.S. Department of Agriculture, as well as it looks like Iowa State University in Ames, Iowa.
Thank you.
It's a pig epicenter.
Very excited about these papers, so I forget to do. I'm very sorry about that. So
they'll forgive us.
13,935 human and swine HA genes, which were sequenced between 2000 and 2021. The PDM09, which is the human pandemic virus, they went from humans into pigs about 370 times after 2009. Right? So
this is over how long is the period again?
It's 2009 to 2021.
Okay, so
I mean,
12
that's limited
12
by
years?
their analysis. If they had more sequences,
Sure.
they could say even
but
more.
that's a
right?
lot.
That's quite a bit. So there's a lot of contact between humans and pigs, and we give them viruses, and more than they give us viruses. In fact, we'll see that later. And then swine to human, they could see 17 swine to human PDM09 cases since 2010. So 370 human to pig and 17 pig to human of this PDM09 virus.
For the
Isn't that
listeners,
interesting?
Vincent, did we say what HA means? That these
No, we
are
didn't.
antigenic proteins on influenza viruses. There's neuraminidase and hemagglutinin. So when people say like H1N1, that's hemagglutinin 1, neuraminidase 1. So there's different combinations of different antigenic proteins. Because a lot of the time it seems obvious to us, but that's actually something that I explained recently to me with my family, and they were like, "Oh, that makes so much sense." So yeah, HA, hemagglutinin, NA is neuraminidase.
So we're using these to track the evolution, but you could also, there are 8 separate RNA segments. You could use others as well.
Yeah.
So some of the interesting facts, so some of the viruses that went into people in 2009, they disappear from people. So specific lineages disappear, but they remain in pigs, and they continue to transmit among pigs and evolve in pigs as well. For example, of one that they found that went from human to pigs, it was maintained for over a year in pigs. These spillovers were found in 22 of 26 states that have swine data. And 17% of all the spillovers spread to 2 or more states. And the states with the most human to swine spillovers were Illinois, Iowa, Minnesota, North Carolina, and Indiana.
That's where the pigs are.
I guess that's
Yeah. There's like no pigs on the
where
West
pigs
Coast.
are.
There's no pigs in the West. Like,
So, Vincent, do you have any idea of how many breeds of pigs
I
there are?
don't know. I have no idea.
I'm looking at the answer right now.
How many
From
breeds of
Wikipedia,
pigs?
it says there are hundreds of breeds of domestic pigs. So my big question is, when this virus first entered pigs, which pig did it go into?
I don't know. It's a good question. It was in pigs for many years before it popped into people, right?
All pigs? I mean, throughout the world?
The ones that they grow for pork, isn't that just one kind?
No. They have hundreds of different, hundreds.
By the way, what's a hog, Angela?
a hog? A hog is just a pig.
Pig.
No, a
So
hog is
Any
just
male or
a
female
swine.
can be a hog?
Swine. I don't think it's sex specific. It's
Okay.
just like
Hog or
the
swines?
yeah.
I don't know
swine,
what kind
hog.
of pig it originally came from, Dixon.
Okay.
I don't know. And also, many of these viruses that went into pigs, they reassort with other viruses that are already infecting the pigs, and you get different combinations. Because influenza virus genome is in 8 pieces, and so when 2 different influenza viruses infect the cell, they can mix and out come mixtures in the viruses that are called reassortants. They could have different properties, in fact.
Right.
And then they see temporal differences. So some seasons there's less PDM09 in people, and they see less spillover into pigs, and that makes sense, of course. In the 2021 season, there was no human to swine transmission, and that was a very low H1N1 season, for example. Let's see, what else is interesting here? The number of human to swine spillovers was an order of magnitude higher than the number of swine to human, kind of already said that. And that correlates with having more PDM09 in humans. So H1N1 and H3N2, which are co-circulating in humans, they tend to vary from year to year which one is higher in number. You know, some years H1 is higher, some years H3 is higher.
I think this year H1N1 is predominant, right?
Predominant, yeah.
Mm-hmm.
So there's one season where there wasn't a correlation between human and pig, and that's 2020-21. That year we had record low influenza activity. Can you guess why?
Maybe another virus.
It might have been all that time we were spending at home.
Yeah.
We all watch Netflix like 12 hours a day.
That's how smart the pigs were. They didn't even come out.
So listen to this. In the U.S. from 2020 to 21, there were 18 H1N1 cases confirmed in the U.S. All right? This is very low.
Yeah.
And they do a lot of flu
that
testing.
18 total?
18.
Wow.
18 total.
That's amazing.
However, they found lots of PDM09 in pigs.
Sure.
It was actually higher than previous seasons. They had 183 detections.
But were they testing for flu? Weren't they mainly testing for SARS-CoV-2? Like if flu at the time would have been
That's a good point. They
tested,
said
no?
despite high levels of testing in 2020-21, they did high
Is that
levels.
true?
I think the flu folks are, testing pigs all the time. This is
No,
part of
pigs,
the
yes.
whole-
I mean humans.
Humans,
No,
too.
I think that
I
people
see.
were. There was a fair amount of testing to distinguish respiratory symptoms in terms of
That's
COVID
true. Okay,
versus
yeah.
flu.
That's true.
So for some reason, the COVID, it impacted human transmission, but not pig-to-pig transmission.
Have you ever tried to mask? Have you ever tried to mask a pig?
Yeah, well, they have an attitude,
Definitely. And a lot
You have
of
to ask them
secretions.
politely, and then maybe they will say yes.
And they
So-
weren't socially distanced. and
Definitely
they weren't
not.
vaccinated.
So
these-
Angela,
Actually,
have
they
a lot
were
of
they are vaccinated, Rich, against certain influenza viruses, the pigs,
Yeah.
for sure.
Okay. Well, actually, that was a question. I was
Mhm.
curious about that.
They are. They have there's multiple vaccines. There's like a bivalent vaccine. I think it's like H3N2 and then H1N1. And then there's also monovalent vaccines. And I believe they're inactivated viral, like whole viral particle inactivated vaccines.
Angela, you said they have a lot of nasal secretions.
Oh yeah. their nose is always wet. It's always like full
That's
of
true. That's true.
secretions. And also, especially if they're sick, because when pigs get sick with influenza viruses, it's similar to humans in the sense that they have coughing, like rhinorrhea. They'll have a lot
Yep.
of like rhinitis.
Hmm.
They'll have a fever. Now, the mortality is really low in pigs, but they do have like these respiratory symptoms, which will increase their secretions, sneezing, coughing, all of
Why is the
this.
mortality rate low? Do we know?
I don't know. I guess it's just, I think it's like 2% or less than 2%.
You mean influenza mortality in
Inf-
pigs?
influenza in pigs?
Yes.
Yes. Sorry.
Okay.
I
'Cause
was
other
looking at the
viruses-
Merck veterinary manual website. The Merck website for vet stuff is the best.
Anyway, so these
That's interesting because 2% mortality in humans would be considered huge,
yeah,
Mhm.
that's right.
But in
Mhm.
pigs,
Well,
for the
2%
reasons
of
that we've
the number of
discussed.
pigs raised in the country.
Exactly.
That's an enormous number.
How many pigs are raised every year in the U.S., Dixon?
About 80 million, maybe more. China raises over 200 million pigs.
But those are the two largest producers of pork in the world, are China and the United States.
That's
And
right.
I
That's
believe,
right.
yes, and then third would be Brazil. Is it Brazil? I forget. But I know China and the US are the two. China's more than the
Right.
United States, though, isn't it?
Right.
It's
Oh, much
hundreds of
more.
millions of
Much more.
pigs. Yeah.
Much more.
Hmm.
That's why they're still interested in trichinosis.
So basically, in that season, the pigs were still transmitting viruses that they got from people from the previous years. It didn't stop because, as Angela said, it's hard to mask a pig, and they didn't do any isolation or any of that. They were just crowded in as usual. Let's see what else. In 20 oh, so they also have evidence in that year 2020 2021 they have 8 cases of swine to human transmission of PDM01 in that year, where we weren't getting it very much. They gave it to a few people as well. All right, so the last bit of information. So basically what we're seeing is that this virus, PDM09, goes back and forth between humans and pigs. And so obviously if it goes into the pig, it can change, it can reassort with other viruses. And they mentioned in the discussion, it could even reassort with H5N1, which can infect pigs and make some new viruses, influenza viruses, which could be dangerous. So the last experiments they do is they get swine PDM9 isolates, and they say, do sera in ferrets that were immunized with human H1N1 vaccines, do they neutralize the infectivity of these viruses that are in pigs? Right? And the answer is it's not as good as it should be. There's a drop in neutralization activity. Okay. These swine strains have a big drop in reactivity with the vaccine, with antibodies induced by the vaccine strain.
When does the drop occur, Vincent?
When?
Yeah, after vaccination. How long does it last before it drops off?
No, I mean not in that sense. If you vaccinate a ferret, if you inoculate a ferret with a human vaccine, and then then in a few weeks, you take the serum and then you try and neutralize these pig viruses, the neutralization titers drop.
Okay.
And they're not zero, but they're low.
Right.
And that's not good because we're vaccinating, and if the vaccine doesn't work against a pig isolate, an influenza isolate from pigs, that's bad, right?
Mm-hmm. So potentially pigs could be a reservoir of a future pandemic virus.
That's right.
Right. So this is, this is like an update on a phenomenon about flu that we've been aware of for a long time, which is that there's interspecies transmission, that the virus can hang out in another species and undergo variation to the point where you've cooked up something that humans no longer have any immunity to. And to me, the update here is that pigs are an extraordinarily robust reservoir of this sort of activity, and that we can pass it back and forth very efficiently. Okay?
And we are perhaps the donor more often.
Yeah. That's right.
Yeah. So
we're giving it the material.
I want to add something to the discussion, which I've just been made aware of by going back to the literature, which I was very familiar with when I was active in my lab, and that is there's another viral disease that Vincent alluded to, which is called classical swine fever. The other name for it is hog cholera, and it's a viral
Oh, yeah.
disease. Now listen,
Mm.
here's their definition. Iowa State University College of Veterinary Medicine. We're talking about this group right now. Here's how they define it. Definition of classical swine fever is a highly contagious viral disease of swine that in its most virulent form causes morbidity and mortality approaching 100%.
Ooh.
But classical swine
So,
fever, because before you said swine flu, which is why we all said that swine fever is a pesty virus, which is like
that's
every
right.
pig gets killed if you have
That's right.
swine
That's right.
fever.
That's right.
It's very, very, very
And
contagious.
if there is one case of swine fever, they kill all the pigs around for 100 miles.
Yes, exactly.
They culled them.
Yes, it's called a
Well, that's a
stamping
polite
out.
word for if you're a pig, you don't use that word.
So
No,
then we have African swine fever, which is a pox virus, right?
Right.
Then a number of
Mhm.
coronas can cause pig infections, lethal, right?
Indeed.
So
Indeed.
pigs are
I would
Highly
call
So
Africans.
close to people in terms of a lot of different things. You can fool a histopathologist by giving them two slides. One has human intestine, one has a pig small intestine. They can't tell them apart.
yeah, really
I would
Cannot
say
tell.
African swine fever is a poxy virus.
poxy.
Poxy.
I like that.
It's actually
I like
its own thing. African swine fever. It's
I
an
like that.
SV virus.
So I have a. Oh, sorry, Rich. Go ahead.
Just one other thought that I had reading this, which is you're not going to do anything about this. Okay? The pigs are going to have flu.
Yep.
The humans and the pigs are going to pass it back and forth. The
That's
best
right.
you can do is understand it. Okay?
Mhm.
And be vigilant. And try and understand the pathogenesis of the influenza sufficiently so that if the pigs are cooking up something that's really dangerous, you can be aware of it.
Yep.
I have to say that one of the ways to understand that too might be gain of function experiments, but
Or we could all just stop eating bacon because that's like the main thing we all
No,
love from
try
pigs.
that.
sorry, Angela. I'm sorry,
It ain't
Angela.
gonna
I
happen.
understand your sensitivity to this. I respect
joking.
it, but bacon.
Oh, I know it's delicious.
Yeah, you can't do
Um,
that to us. You can't, please.
So
Okay, so-
as Rich said, what they say in the papers, we need to do a lot of surveillance to see what's in pigs and characterize it against sera induced by vaccines. And we need to do more of that. Yeah.
Which is so much easier because we say the same about bats, which is extremely hard, other animal
Yeah.
reservoirs, wildlife reservoirs. But our pigs are literally in our intense farms. We have them. We
Yep,
literally farm them. There's no excuses not to do a better job of surveillance. Now, one
Right.
of my questions is, which I'm not actually aware of this answer, and I just thought of it. So we know that avian influenza can spill over into pigs, but then if it reassorts in this pig, do we know of any cases where it can spill back into birds? Or
Oh, that's
has
an
that
interesting
not
question.
been documented? Like, do we know if they can then?
I can't
would
imagine
think-
that it wouldn't happen, but I don't know any data.
It's a good I think so, but I don't know of a good example. Yeah, that's a good question. Should
Because
look
then, if
into that.
imagine from like the birds to the pigs, back to the birds, then back to
Yeah.
us. There's
Well, certainly the avian influenza, some of those genes went into the 2009 in pigs, but reverse
Mhm.
is a good question. I think
Mhm.
I want to look up. If someone's listening and knows, let us know. But I think we
Right
should look
now.
it up.
Okay, quickly Google. Yeah.
Well, if we were live, it could be right now. Yeah. We could do a live stream of this like now and then. That would be fun.
Mhm.
That would be fun.
Because all of you have been on the live stream, right?
I have, yeah.
Sure.
Brianne has,
Yeah.
Dixon has, Angela, you have, right?
Yes.
Yeah.
This week I had Cindy.
Awesome.
Nice.
hmm
So there's a hope for a vaccine here, but only if you
What do you
found a
mean?
universal influenza vaccine, right? Because it keeps varying, and all of these variants
A universal would be good. In fact, that was one of the papers that I had
Yeah,
lined up as a
exactly,
universal,
exactly.
which we may do another time. Yeah.
But the danger of this vaccine, if it didn't work so well, but it worked enough to lower it from 1%, 2% to 1%,
Yeah.
it means that you could never test for the H1N1 virus anymore because they'd all be positive. Pigs only live about 6 months.
They live 6 months because we killed them, right?
We do.
Yeah.
What's the lifespan of a pig, Angela?
Oh,
I
it's
think they can live like
25
20
or 30
years.
years,
Yeah.
right?
Like over 20 years.
That's right.
There's a
If they're
line in Cannery Row where if you haven't read Cannery Row, this ought to be
It's a
week.
great story.
It's amazing. Okay, where the bums go on a frog hunt to help Doc out by supplying him with frogs, and it of course goes bad. And on the way out to the frog hunt, they roadkill a chicken and take advantage of that to sit and roast the chicken. And during their conversation, somebody asks, how long does a chicken live anyway? And the answer is, nobody knows.
Nobody knows. All right,
well,
let's do some picks. Dixon, what's up?
Picks. Oh, I love it. Pigs, not pigs. Picks, right?
Picks, yep.
So my pick is, as soon as I get it up here, because I've closed my window, unfortunately. That's okay. I can open it really quick.
I heard this on NPR yesterday.
Yeah. The death of a wonderful, wonderful person who I listened to ad nauseum whenever I could. His name is Peter Schickele, and he's also known as PDQ Buck. That was his moniker for hiding behind what was obviously a genius in musicology. And he would give forth the most amazing, off the top of his head, explanations for musical concerts, writers. He knew all the literature. He knew all the writers. He knew all the composers. He knew everything. As far as I was concerned, he knew everything. And I just absolutely loved him. And he was as funny as hell. Funnier than Victor Borge even. You took the picture down, but I had a picture there.
No, I didn't take it. It's still there.
Is it?
Yep.
Yeah.
Oh, I'm sorry. I didn't scroll down enough. That's right. This is one of his least humorous pictures. And he was a remarkable person. No other person ever did what he did. Tom Lehrer, Victor Borge, they didn't come close to him. He was encyclopedic in his knowledge of music. And this concert for horn and hard art, I'm sorry that it copied itself on each one of the pages for our show notes, and I guess you'll have to take it down. But horn and hard art, I don't know if those are people out there that don't know what that is. It was a restaurant in New York City that you could walk into, and it had windows, like they looked like mailboxes.
Mm-hmm.
And you would go over to a window, and you'd look inside the mailbox or window, and you'd see a slice of lemon pie, or you would see a turkey dressed, or you would see a drumstick, or you'd see a pot of a plate of peas or something, and you'd put a dime or a quarter, or in those days it was pretty cheap, and the little window would open, and you'd reach in and you'd grab that piece of food, and you'd put on your tray, and you'd build a meal like that. And so Peter Schickele wrote a concert in honor of the Horn and Hard Art restaurant. Concert for horn, okay, and hard art. Where did that come from?
That's funny.
And the way you should listen to it, it's charming and it's humorous, and it requires a little bit of knowledge of music as well. And that's interesting. By the way, I noted that he was 88 years old when he died, and his last picture was taken of him sitting on a piano.
Yeah, there you go.
So he lived his music. He was an incredibly approachable person. That's all I can say.
Thank you,
And I
Dixon.
loved him.
Thank you. Angela, what do you have for us?
So mine isn't like a specific thing that happened this week. It's more so I wanted to have kind of a discussion and to know your opinions regarding ChatGPT, and more specifically ChatGPT 4. And if any of you have used it, or what your opinions are surrounding it, and if you like it or not. Brianne, if you've or Vincent, that you both teach still, if you allow it to be used in the classroom, and if so, with what limitations and how. So there's like a lot of, I just think that it's a really, we're going to need to just accept it because it's here and it's not going anywhere. And like establishing kind of ways, tools we can use it to benefit the classroom, as opposed to just saying, oh, you can't use ChatGPT because everybody's going to use it. That's not going to happen. Like every student in the world is going to use it. And now with ChatGPT 4, which Dinesh, one of the PhD students in my lab, he was doing like a trial for a month. I think it's $20 for a month, for a monthly subscription, which is very cheap. And the things that he was doing with it was crazy. So you just have to learn from what I saw. I'm terrible at putting in prompts, but the better your prompts are, obviously, the better it understands you. But we literally input like an RNA-seq data set and then asked it to create like volcano plots and all of this. And it was okay at doing it, like a bulk RNA-seq data set, but it more or less, it was because our limitations, like we didn't give it enough prompts for it to do its job properly, but it was amazing what it did. And then it would reroute, because obviously it has these hallucinations too that you have to take into account, but it would kind of get stuck, explain to you where it was troubleshooting, and then reanalyze and then start again, and then it would spit out more data. It's crazy. So yeah, anyone's opinions on ChatGPT 4?
So I haven't used ChatGPT 4 or any of the previous iterations to look at data in the way that you mentioned about the RNA-seq database, but I have talked about it a lot in thinking about teaching. We have a lot of faculty sort of sessions about it, and I would teach writing intensive classes in a lot of my classes, so it actually comes up in a lot of my classes. This past summer, we had to actually take our prompts for our writing class and put them in and see what types of things we would get, and we were all really sad by the answers, and we ended up spending a lot of time thinking about how to write prompts that wouldn't necessarily give as great of an answer, that wouldn't be as amenable to a student writing with ChatGPT. In general, it's part of a field known as generative AI. And so it becomes really hard to ban ChatGPT because you can think about some of the other types of generative AI and places where they kind of bleed into having similar things. So any of your predictive text or predictive spell check is also generative AI.
True.
As is Grammarly in some cases. And those are all things that we frequently let our students use. And so we spent a lot of time thinking about both ways to discourage use but not outright ban it, and ways to talk to our students about it as a tool that perhaps they have to cite. Like at the end of this paper, I use spell check. I got some ideas from Grammarly, and also I used some really good hot chocolate. And so that was actually like what we were talking about, is trying to talk about our students always having an idea of sort of acknowledgements of things that really helped them write the piece. There are some really useful ways that we learned to use it in teaching writing. So there are some super useful things. There are also some things that are awful. I would recommend you ask it to write something with citations, or to give you a formatted citation, because it will give you beautiful citations, all of which are messed up.
It's true. But 3.5, it's terrible, but you can actually, so in 4, because I also had the same problem, or it would like just hallucinate and make up citations that were completely.
It totally hallucinates them.
None of it
would
makes
be
sense.
Like Racaniello, and then 2023, and then a title of a paper written by Angela and a journal that Rich has published in, all kind of Frankenstein together.
There are like, I forget what they're called, but in ChatGPT 4 now you can input like channels almost, or like they're kind of like users within that are more like precise, and some of them for referencing specifically. Another person in my lab, Connie, her husband is like deep in AI and machine learning, and I forget what it's called specifically because I don't actually use ChatGPT 4. I've only used it twice, but there's more specific like subsets within ChatGPT 4 that you can actually use that'll give you a more precise like more specific to in the context that you're looking at. Like if you're looking at like references for papers or like for science specifically, or if you want something for like, I don't know, coding, like for R or all of that, they're like more specific, but you have to pay for it, obviously. So, and even for data, so that's another thing that's like can be dangerous. So obviously the data set that we put in was already published, something that's already published, because then you have to think that like any data that you give it, it is then going to use to generate questions and ideas for other people that are then coming in after, unless you have it locally, which I don't really trust how local it is, because you can do that. But I'm not sure how local it actually is. And if that information, I mean, obviously, like I'm sure big like companies do have local and they have firewalls, or I'm not sure how they use it, but me in my home using ChatGPT-4, I would never put in data or even like that's been unpublished, because then it'll just use that, whatever you give it, to then answer other people's questions and make its own links and all of this stuff, right? So I think it can be dangerous in that sense. But yeah, I don't know if anybody else has any ideas.
So
or
I
has
have
used
no
it.
experience with this, but I have an idea that I'm sure has been thought of 1,000 times. How about a class, if you're going to, accept that people are going to use it no matter what? How about a class where basically a fundamental component of an assignment, or maybe all assignments, is to get a Chat GPT essay, and the assignment is to edit that.
That is actually the number one thing that we learned in faculty development, is that it can be really good and for things like generating sample essays for students to work on, or to have a student generate something and then have to fact check it or have to think about what's good and what's bad. That was the number one most commonly made recommendation.
Yeah, it seems to me you'd learn about prompts. You'd learn about how accurate ChatGPT is, and you'd also learn about the subject if you're required to do the edits, and maybe even what you have to turn in is the original essay, the edited essay, and then the Word doc that shows the edits that you did.
That's exactly the direction that people are going with this.
So with this program, if they handed you an electronically generated document, like an email or an attachment, would you know by reading it whether it was manufactured by ChatGPT?
You can use ChatGPT. Ask ChatGPT, right?
They have software that can detect, but the software is ChatGPT is getting better and better at evading the software. And now some of that software tends to pick up language written by non-native English speakers. Because it sort of also is a little formulaic. So generally, no, you cannot. So generally, no,
yeah.
If I were in charge of this world, which I'm not,
you're not,
but I would make it a requirement for every document ever produced by ChatGPT-4 to say at the bottom, this document was generated by GTP-4.
But that's not in their best interest. Why would they do that?
Not only do they not do that.
No, I know they don't do that.
Not
I'm
only
saying,
do they not do that, it is not saved anywhere.
Exactly.
So if I, as a professor, said, hey, I think you used ChatGPT, I can't go back in the history of the internet and find the document and be
I would change all of that. I would change.
And it will never, Dixon, it will never spit out. Brianna and I can both ask at the exact same time, the exact same question, and it'll give different answers. So if I ask for an essay, a 500-word essay, no, but I mean, it'll be very different. And then even if it's
but you can't tell, right? Because there's no identifying signature that this was generated by AI.
But that's what I mean. And you can also ask for something. So a lot of the time, some students, they're not as smart with their prompts. I've seen it already. I'm like, this is freaking ChatGPT, because it comes up and it's so like eloquent and like beautifully spoken. But then you can ask ChatGPT to have more casual speech, or you can say, oh, can you rewrite this in a more casual manner, or in a more like, you can say literally anything and it'll rewrite it for you. So there's no way for us to actually know if students are using it or not. Like there's
how did they
just
catch
no
the president
way.
of Harvard?
Well,
how did they catch that woman?
That was not a ChatGPT issue.
I'm sure of that. But there must have been some machine that read it and said, "Oh, I've seen that before. It's in another document."
There's been software around forever that will detect plagiarism.
Yeah, but Grammarly will do.
Of course.
So why can't this fall into that category?
Because it's not saved, like
each
Brianne
one is
said.
unique.
It's just
Yeah.
gone.
That's
It's
it.
gone. You close, like it's not saved on a cloud. Imagine how much like space we would need to save every single prompt and
Yeah.
every answer that it gave. Like, and also even with ChatGPT 4, you have a maximum of 30 prompts per 3 hours. So you can't just like unlimited, it takes a lot of resources for ChatGPT 4 to work, right? So you can't just like ask it constantly questions, or prompts, I should say. But
Now you know why the world is
It's-
afraid of AI.
So I don't use it a lot. I haven't used 4. I've used earlier versions, but I sometimes when I try to make a definition, I use it for help.
Mhm.
Right. And I always say, please, in one sentence, define this, because if you don't, then it goes on and on
Mhm.
forever. And it's not really useful to have a whole paragraph so if you want to say, what is a virus? Tell me what is a virus in one sentence. And that can be better, I found.
Exactly. Yeah. The more specific you are with your prompts, like
Yeah.
if you're very vague with the prompt, it'll be just as vague because it doesn't know what you want, right? It has no idea.
But I
Um,
really like asking it, who is Vincent Racaniello? And each time it gives a different
hallucinates.
answer. And then it makes up some freaking papers, as Brianne said, that I've never published. And I said, where the hell does this come from?
But no, it's getting better. It's getting better with ChatGPT 4.
But-
3.5, not so much, but
I think it is part of our creative process, and it's going to be more and more. And I don't have a problem with people using it to frame an essay and then fix it themselves. It's like using AI to make art, and some of it looks amazing, right? As long as you say this is AI art, I think it's all part of
As
what
long
humans
as you
do.
say, that's the point. As long as you
Yeah, I
say.
think you have to
Yeah.
acknowledge it. Don't say this is my writing. And, you have
That's
some good questions here. I mean, one of our concerns at Columbia is that they use it to answer exams. But in my case, my exams are timed, and there are just too many questions for them to put each one into Chat GPT. So
Yeah,
Mhm.
that's like having an open book exam.
Yeah, I mean, I tell them,
Mm.
look, you have an hour and it's open book. You can't look everything up. You need to
Right.
know stuff.
Right. Exactly.
But it's going to get better and better. It's going to be in the classroom. Yeah, writing essays. Well, you say, will it not advance if we no longer update blogs? But we will always update
But it's
what's
true.
online, right?
If you think about like coding, for example, the amount of
Yeah.
people that ask ChatGPT now for code, which normally you would just go to like blogs and like all of these other places, you would find that and
Yeah.
people are updating it. But if people stop updating those,
Yeah.
then it won't have training data and then it won't progress. So that's like one of the, I'm
It's a
not
good,
sure,
interesting
obviously people
question.
that are smarter than me have thought of this,
Yeah.
but we were discussing this recently at a lab meeting. Like we were just discussing, my PI said, let's have a discussion on ChatGPT 4 because it's here and it's here to stay. And we need to
Yeah.
discuss like how we're going to use it, which is where she brought up, don't put in any data into ChatGPT 4 because there's like, then it's part of, just you're training it on data that hasn't even been published yet. So it could be like kind of like getting scooped via ChatGPT 4, which
Yeah.
it could be. We don't know, right? So
Your last question, do you think AI will become conscious or sentient? Yeah, of course it will. Just look at Ex Machina, right? Did you
do
see
you
that
think
movie?
though?
Yeah.
So, but
Yeah,
like
absolutely.
Yeah, I've seen that.
sentient,
It's a good movie.
I don't agree. I think the way we define our sentient, like us as sentient beings, like feeling and conscious, like what human consciousness is, I think
Yeah.
it'll be its own form of artificial consciousness,
Maybe.
but it'll never be, in my opinion. I think saying that is like undermining how complex the human brain is. But
Mm.
and how like we can just like create, well, I don't know. I think it's I would say no. In its own way, maybe, but not.
A few episodes ago, I
consciousness.
picked the Murderbot Diaries, right?
Yeah.
And go read some of those because that's
right.
that's essentially about sentience in
Yeah.
artificial intelligence.
It's
Mhm.
very, very good. I like that discussion, Angela. Thank you.
Yeah. No problem.
Probably-
It was
probably do it
inspired
again.
by a recent lab meeting, but yeah, I
We could
think
do
it's
it. It would be interesting to get some expert on and get it
Yeah.
and have us and talk about it in the context of science. That would be fun.
it's true, yeah, because I'm like I know nothing. I
Yeah,
know
neither do
barely
I.
anything, but somebody else that has more,
know
like I know
less.
very little. Somebody that
I
has
would,
like an understanding, it would be
if
great to have them
anyone
on.
knows someone who could have a discussion with us about AI and science, that would be great. Tell us who it is, because I don't know. Brianne, what do you have for us?
So I've been looking at some resources that I had for teaching, and I came across these links that I have put aside that are two TikTok videos about RNA vaccines that I love, and I feel like I share with a lot of people. I Googled and saw that I hadn't picked them. It looked like, and I was shocked because I shared them so often. So these are one particular creator. He actually has 10 different vaccine related videos that show in one case how an mRNA vaccine works, and in another case why one might need 2 doses of a vaccine. He has others on other aspects of coronaviruses and different vaccines. And for a layperson, and sort of for a very simple description of how vaccines work, the idea of, the mRNA coming into the cell, giving a message to the ribosome, and then getting degraded, and then the production of this small thing that is a part of the bigger virus and the attacking the virus. All of that, I think, seems to really resonate with a lot of the lay people who I've shown these to. My students think it's hilarious that I know what TikTok is, and we'll show them some of these TikToks from time to time. And so if you need to explain some of these things, I recommend these videos a lot.
Cool.
Brianne, these links aren't
Those are
working for
cool.
me.
They're not?
No. Am I alone in this?
I'm always afraid to click on them in case they make noise.
No, mine worked.
Yeah,
It's working.
So when I click on it, it takes me to TikTok, and I have to click continue as guest.
Yeah, it works.
Says it's redirecting me, and then spins its wheels for a while, and
I've got it.
Okay,
It's a local issue.
I'll figure it out.
Rich, what do you have for us?
This actually turns out to be relevant to this episode. On one of my late night, which for me is probably about 9:30, ventures down the internet rabbit hole trying to find the end of the internet, I was, for some reason, musing on sort of the coevolution of just humans in general and diet. Okay, and I was mostly, I think, interested in the relationship between meat eating and evolution. Like, when did this happen in human evolution? How much of an impact did it have on our evolution? How close were we to where we are now in our evolution when meat eating happened? And I'm particularly interested to know whether there, and I don't have an answer to this from this article, but whether in the evolution of us as meat eaters, there has been evolution in metabolic pathways that I don't think, I mean, obviously you can still be a vegan or a vegetarian, so we aren't locked into actually having to eat meat, but has there been evolution in pathways that somehow make it so that eating meat is a more efficient way to get around, or whatever? I mean, clearly we don't have a meatless equivalent of scurvy, okay? But I wonder about whether there's been any sort of metabolic pathway evolution. At any rate, in the course of that, I came across this article, and I've actually communicated with the author some to try and refine my understanding of things. The article is called Evolutionary Basis for the Human Diet, Consequences for Human Health by P. Andrews and R.J. Johnson. And Andrews is from the Natural History Museum at London University, and Johnson is from the University of Colorado Anschluss Medical Campus in Aurora, Colorado. And I'm sure this is one of probably thousands of articles that are out there. But one of the things that I learned from this is that critical to the, is that our very distant ancestors, and this was before we were even Homo, were frugivorous, right? So, which is essentially vegan. We lived on fruit, okay, and things that you could pick up off the ground, and that skipping over a lot of stuff, a big transition point was the controlled use of fire. which happens just really at the very beginning of the emergence of Homo, okay? And was significantly before Homo sapiens. And that, in fact, the use of fire to cook things and do other things as well had a huge impact on our evolution. And so essentially meat eating happens very early in the, or actually significantly before Homo sapiens. So there's a lot of evolution that has gone on since we started doing this. And I still don't have an answer to the question as to what, maybe somebody out there listening to this can give me some more references as to what the impact might have been on our metabolism. Not metabolism rates, but pathways.
Yeah.
At any rate, there you
It's
go.
very cool. Very interesting topic. I like this. Yeah.
Yeah.
Sorry, I
wondered.
stepped out for a moment, but did you
Don't worry.
mention that meat eating triggered big brain development?
Yeah.
Yes, that's
Okay,
part of
okay,
this
great.
article. I didn't actually
Okay.
mention it, but
Yeah,
that's
it's
part
part
of
of
this article. There's
So I think once we got a
evolution of
taste
both the
for it, we
gut and
couldn't
the brain.
lose it.
Well, and
Exactly, because if we don't have vitamin B12, which is mainly from animal sources, then we get like different sorts of, what is it, megaloblastic anemia. And there can
Spina
be
and all kinds of
lots
other
of
things.
different
That's right.
deficiencies. Yeah.
You got it.
one of the points that's made here is that the brain is a very energy expensive
Yep.
organ. And so
Yeah, it takes 25%, right?
And so, in fact, in order to facilitate, in an evolutionary scale, brain development, meat eating is important because it
Yeah.
provides a lot of energy quickly.
Also, the speed at which you get your calories. You know,
Mhm.
when you kill something and you go to eat it, there are lots of other things out there that would like to help you eat it and perhaps eat you at the same time. So we learned in our beginning, at least as far as what I've read, we didn't eat the meat. We waited for the omnivores and the carnivores to finish the carcass, and then we casually walked over and broke open the bones and ate the marrow. And we survived a lot with that to begin with. And then the hyenas, which didn't evolve in Africa, but rather in Europe, as soon as a land bridge was made between Africa and Europe, the hyenas invaded Africa. Hyenas can eat bones. And so the carcasses were suddenly not available to us. And I think I've read anthropologist Johnson and lots of others that have speculated as to that's the point in which we started to develop weapons and defense systems and clans that hunted together and that sort of thing. It's complicated though, Rich. It really is, isn't it?
Yeah.
I mean, it's not a simple story.
And it's fascinating. I got a nice
Mhm.
figure here that has Homo habilis showing up 3 million years ago at about the same time as being omnivorous among our ancestors, okay?
Indeed.
And Homo sapiens not showing up until 800,000 years ago. So there's 2 million years of evolution,
That's
okay, of us being omnivorous before we even get to Homo sapiens.
Right.
Hmm.
Cool. That's great. Thank you, Rich.
This is what happens to a science nerd
Yeah.
when the TV
No, it's good
gets
stuff.
turned
Good
off.
stuff.
My pick is this year's version of Virology lectures,
Oh, nice.
which just started this week. There's no link right now for you guys, but I'll put the first lecture up on YouTube tomorrow. So there'll be 25 lectures of introductory virology. We have 2 listener picks who both picked the same interview with Dixon in New Yorker magazine. So there you go, Dixon.
Thank you.
The New Yorker
Yay.
interviews Dixon wants our cities to be like forests.
Indeed.
It's a great article.
It's very cool. And I have I get the New Yorker. I don't know if it's in the print copy. Do you know, Dixon?
It's not. No, it's an online article.
Okay. I have a subscription so I can go online as well.
You can do. I don't, so I had to get my wife to do it.
I get the
When-
New Yorker because that was my perk for being a subscriber to NPR. How about
That's
Oh,
that?
how wonderful.
So we have 2. One is from an anonymous listener and one is from Jeff. So thanks for that. And that will do it for TWiV 1081. You can find the show notes at microbe.tv/TWiV. Send your questions, send your comments, send your pics to twiv@microbe.tv. And if you enjoy our work, please support us. Go to microbe.tv/contribute. Dixon Despommier, Racaniello.org, thelivingriver.org. Thank you, Dixon.
Marvelous show. Keep up the good work.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Brianne Barker's at Drew University, BioProf Barker on Like Blue Sky and Mastodon and all that stuff. Thank you, Brianne.
Thanks. I learned a lot.
Angela Mingarelli's at McGill University, Immune Vet on X, right, Angela?
Yes. I don't have other social media anymore, but yes, X.
Thank you, Angela.
Yeah, this was great. This was so much fun. I hope you guys have a great weekend.
I'm Vincent Racaniello. You
You
can
too.
find me at virology.ws. No, actually, I don't write there anymore. You can find me at microbe.tv. I want to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.